So, I am sure that you're aware that in high school, you were introduced to the concept of mitochondria being the powerhouse of the cell that helps to make energy for the whole body. Now, you can imagine that if the mitochondria doesn't perform at the level that it's supposed to or expected to, then your body will lack the energy from this powerhouse, and this will manifest as symptoms. If you remember the introduction of this presentation, I talked about the 6 different ways that vitamin K2 can impact Alzheimer's disease, and one of them was mitochondrial dysfunction. Now, I have already talked about beta amyloid and neuroinflammation, and now I'm going to talk about mitochondrial dysfunction. However, I am not going to talk about it in isolation. And as you can see, this is kind of like a 3 plus 2 plus 1. So here, the first element that I talked about was the beta amyloid. Second one was the neuroinflammation. And the third one is going to be mitochondrial dysfunction. However, the studies referenced in this section kind of they impact all 3 different mechanisms. So I'm not going to talk in isolation, but rather how it impacts all of those 3. So, uh, from decades of research, we know that mitochondria doesn't work properly in Alzheimer's disease. And this is really shown through decreased ATP production and increased reactive oxygen species. If you're ma- So think of ATP as kind of the single common molecule used by your whole body for energy. If you're making decreased ATP, essentially your body has less energy to work with, leading to eventual faults such as the symptoms in Alzheimer's. So when we're talking about mitochondria being the powerhouse of the cell, that's mostly because it produces ATP, and the ATP is the true powerhouse of the cell, but it's produced by the mitochondria. So, furthermore, in Alzheimer's disease, as I talked about, there's also increased reactive oxygen species in the mitochondria. So, this is essentially the gunk, which impairs the performance of the mitochondria and also leads to damage in other parts of the body. Reactive oxygen species is something that we don't want, and we want to reduce it as much as possible. So, what is interesting is that in the same study that I presented for neuroinflammation, the researchers also found another finding with the glial cells. Remember that I said that there were 2 support cells for neurons, astrocytes and microglia, and they're both classified as glial cells. But in this case, the researchers use astrocytes of glial cells. So I'll just talk about the same study again I talked about before. But first of all, the model that they use is the hypoxic Alzheimer's disease model. So this one of the ways that Alzheimer's disease can be modeled in a mouse model is by causing hypoxic or low oxygen conditions. And by causing hypoxic conditions, this leads to an inflammatory cytokine response, which leads to neuroinflammation, which is one of the signs of Alzheimer's disease. So remember back to that diagram that I talked about, how like 5 different factors can basically go down the flow chart and lead to neuroinflammation by increasing the levels of inflammatory cytokines. Well, this is what I was talking about. So, by pre-treating the Alzheimer's disease mouse model with vitamin K2's MK7, they found that MK7 decreased astrocyte glial activation and the inflammatory cytokines interleukin 1 beta, tumor necrosis factor alpha, and interleukin 6, as discussed previously. However, they also found a very interesting result. So vitamin K2 help these mitochondria to increase their ATP production and decrease the amount of reactive oxygen species. This essentially means that it helped to reverse the symptoms of Alzheimer's disease on the mitochondria, helping them to return to previous function and also become the powerhouse of the cell once more. So, in the one second. So, in another study, this time using flies, they had genes that cause them to develop Alzheimer's disease, and vitamin K2 treatment found a number of interesting things on these flies. First of all, it decreased beta amyloid. It also helped to increase ATP production in the mitochondria. It reversed negative mitochondrial changes in these flies, and it even amazingly prolonged the lifespan of these flies. Furthermore, it was found that it activated and increased the levels of autophagy. So, what does this mean, and what is autophagy? So, if you look at this person over here, he's Yoshinori Ohsumi, and in 2016 he found the Nobel Prize. He got the Nobel Prize in physiology or medicine. So, and he got this for discovering the mechanisms behind autophagy. Autophagy is essentially your body's way of removing garbage proteins. Think of this as getting rid of the old to make way for the new and better. If we don't have enough autophagy, then proteins that don't work the way that they're supposed to stay around longer and interfere with function, such as Alzheimer's disease. Now, let's go back to the fly study after being introduced to autophagy. Oh, autophagy. So how do we know that? How do we know that this autophagy was actually increased, and what does this mean? So because of Yoshinori Ohsumi, we know that the mechanisms. We know the mechanisms of autophagy, which is in essence what genes are involved in autophagy. So, in this fly study, vitamin K2 increased the expression, or in other words, increase the amount that these genes are active. This results in more autophagy, not just in neurons, but in a lot of different cell types. Activating autophagy is well documented in the literature to decrease beta amyloid and improve cognition. I hypothesize that perhaps this means that autophagy is clearing away the beta amyloid, but regardless, the ability of vitamin K2 to have such profound effects brings about it the possibility to be a new therapy for Alzheimer's disease. And it is very disappointing to see that there are no human clinical trials to see its role in Alzheimer's disease with different dosages and different durations of vitamin K2. And I believe this is something that should definitely be researched.